Morning, everybody. In today's video, we're going to cover vision. Now, for Chapter 5, Sensation, what you have to know and be able to explain is how each of the senses takes energy and then converts it to neural messages. So today I'm going to go over vision, how our visual system works to take energy, light energy, and convert it to neural messages. All right, now, first of all, the term for the converting energy, all right, to neural messages is known as transduction. So whenever energy is converted to neural messages, that's known as transduction. Now it takes place with every single one of your senses. In vision, the stimulus input or the energy is light waves. Now, you know about the electromagnetic spectrum, all right? And basically the light energy that we can pick up or detect with the human eye is visible light or white light. So we can't detect everything along the electromagnetic spectrum, but we can detect visible light or white light. Now, in vision, the length of the wave or the wavelength ultimately is going to determine color. Okay, now if you look at the visuals on the screen, on the bottom left, you'll see U color. So a short wavelength, so if you look the top of one wave to the top of the other in blue, that wavelength is much shorter than the top of the wavelength to the top of the wavelength in the next wave in red. So the length of the wave ultimately dictates color. And to the right, you can see different colors and the wavelength. So you must know that wavelength is the distance from the peak of one wave to the next, ultimately determines hue or color. Now, when we're talking about the amplitude of the wave, we're talking about the height of the wave. So as you can see, there's two examples with smaller amplitude and greater amplitude. And in vision, the amplitude will give us intensity or brightness. So you can think of a bright color, say bright fluorescent pink, versus dull or pastel pink. So that has to do with the amplitude of the wave. All right, now vision, I'm going to run through it. Here you have a diagram of the eye. Okay, so the candle is what the eye is focusing on. Now, the thin outer covering, all right, protective covering of the eye is known as the cornea. What does the cornea do? It takes those light waves or energy that hits the cornea, it bends those rays inward or into the eye. All right, so it protects and bends the light waves in. Now, once it passes through the cornea, the next thing that it will pass through is the pupil. Now, the pupil is the small adjustable opening of the eye. Think of it like a camera shutter. So it will adjust to let more or less light in. Now, what controls the opening of the eye, all right, or the pupil, is the iris. All right, tiny colored portion of the eye that will control the size of the pupil, the opening and closing. Think of it almost like a camera. Now, what you have is you have the cornea. Behind the cornea is the pupil. Lying behind the pupil is the lens. All right, now the lens, what happens as the image and the light energy comes in, it passes through the cornea, it passes through the pupil. It then passes through the lens. The lens flips and inverts the image and projects it onto the retina. Now to do that, the lens must accommodate it. So it's known as accommodation. It changes its curvature so that it can project the image directly onto the back of the retina, the light sensitive surface. So think of it almost like in class when I use the overhead projector. When I put something on there, I have to adjust it. All right, it moves up and down, and so that I can adjust the image directly onto the screen and it can come into focus. Well, it's the same thing. Through accommodation, the lens does the same thing. It takes the image or the light waves and projects them directly onto the back of the retina. So you can see all that going on. Here's the candle's in the back. Some things to make note of on this slide is you see there it's pointing at the fovea. The fovea is the point of central focus in the eye. So that's where ultimately the images will be projected. So you can see the candle is flipped and inverted and projected right onto the back of the retina by the fovea. Now the blind spot, it's a little misleading somewhat because of where the line is going to. But if you look, the optic nerve enters the eye. Okay, and right where it enters the eye, there are no receptor cells, which we'll talk about, rods and cones. Now, since there are no rods or cones, that creates a blind spot in our vision. So we have a blind spot in both eyes. I'm going to demonstrate that in class to you so you can see, actually become aware of it. All right. So the fovea is the area of central focus. The blind spot is where there are no receptor cells, rods or cones, therefore you have no vision in that one area of the eye. Okay, and you can see the optic nerve. What does it do? It relays neural messages onto the brain. All right, now we said the lens changes shape. That's accommodation so that it can project the object and the light and the image directly onto the retina. Now the retina, you must know the retina is the light sensitive surface of the eye. Okay, now what is embedded in the retina are rods and cones. Rods and cones are the photoreceptor cells. When the light waves hit the rods and cones that are embedded in the retina membrane, stimulates a reaction, which then generates a neural message. So as soon as the light waves hit the rods and cones in the retina, that is where transduction takes place and a neural message is generated. Okay, now, then where does the message go from there? So the rods and the cones are activated. Then the information, the neural messages, are relayed to bipolar cells more often than not. So bipolar cells. Now the bipolar cells relay the neural messages to the ganglion cells. The ganglion cells wrap together and they form the beginning of the optic nerve. So think, the neural message, which is generated in the retina by stimulation of the rods and cones, the neural message goes from the rods and cones to the bipolar cells to the ganglion cells, which form the beginning of the optic nerve. Now the neural messages are being relayed along the optic nerve. They're also being processed en route. Now, as they go along the optic nerve, where are they going next? They are traveling to the thalamus, or the sensory switchboard receives all sensory information except for smell. Once it reaches the thalamus, it is visual information, therefore it is relayed to the visual cortex, which is the back of the occipital lobe. Okay, on this slide you can see a little bit, there's the membrane, there's rods and cones that are embedded deep within. Now if you can see, the cones are in color, the gold, and the rods are the bluish color. Now, as you can see, there's many, many more rods than cones. It's a 20-to-1 ratio of rods to cones. The other thing you should make note of, as you can see, cones have their own distinct bipolar cell. So if you look, they link up with a bipolar cell, and it's the cone and the cone alone. Now, on the other hand, if you look, several rods transmit to a singular bipolar cell. And then beyond the bipolar cell, you see the ganglion cells, and then they go out and the beginning part of the optic nerve. Okay, that's important information, and you'll see why in a minute. Now, as you can see in the lower part of this, it's the neural message. It's just reiterating what I went over. Rods and cones transmit neural messages to bipolar cells. Bipolar cells relay the message to ganglion cells. Ganglion cells form the beginning of the optic nerve, and now the message is on the way to the thalamus, which will then relay the visual information onto the visual cortex. All right, now you need to know a little information about the photoreceptor cells known as the rods and cones. Now, cones, they look like cones. Where are they centered? In the fovea. So color vision is best in the center of your vision. As you move towards the periphery, you have more trouble detecting color. All right. Now, if you look, it also says fine detail. Once again, remember the cone had its own singular bipolar cell. All right. So that's why it excuse me, enhances fine detail. Now, a very easy way to demonstrate this, I want you to look at your textbook and stare down at your textbook. You'll look that the words in your center vision are sharper, crisper, clearer. And as you go towards the periphery, they become more blurred because in the periphery are your rods. So cones detect detail much better. That's why it's sharper in the center, because that's where they're located. And in the periphery, you have rods, which that makes it a little bit blurrier. All right, so that's cones. Remember, cones, color, center. Cones, color, center, and also fine detail. Now rods, on the other hand, there's larger numbers, so they work more together. So they're in the what? Periphery, or the edges of your retina. Now, what are they really good at detecting? Black and white. All right, also gray. Now, how you can see where your rods are at work, see there's a larger number, they work together. Think about at night, you get in bed, right before you get in bed, you turn out your light, you get in bed. Now, it's pitch black, you can't see anything. Now, if you wait about 10, 15 minutes, sometimes a little bit less, what you'll notice is you can make out every single thing in your room, your dresser, your chair, your books on the floor. They're all different shades of what? Gray. All right, that's because your rods are at work in dim light. So you can see things, but if you notice and you think about it, there's no color. It's just what? Grays, shades of gray. That's your rods at work. All right, so then you can see 6 million to 120 million. All right, we talked about that. You can see location, where they are, center versus periphery, um, low light, so on. Okay. Now, as we move along, this we already talked about, the chain reaction. So once neural messages are relayed from the optic nerve, where are they going? The optic nerve is carrying those neural impulses from the eye to the brain, specifically the thalamus and visual cortex. Now, the blind spot, I'm going to demonstrate that in class, but you have that blind spot because think, there are no rods and cones where the what? Optic nerve enters the eye. Therefore, that light energy is not being converted to neural message, therefore you can't what? See anything there. All right, fovea we said is the central point of focus. All right, blind spot demo, I'm going to do this in class. It doesn't work well on the screen. All right, now when they're talking about visual sharpness of vision, they'll talk about acuity, visual acuity. So a perfect vision 20/20, okay, sharpness of vision. Now that test that they give you, so you know the vision test has the chart with the giant E at the top and then the letters that go all the way along. That's known as the Snellen chart. So know that just in case. They have been asking that lately. So acuity is sharpness of vision, 20/20. The chart that actually you are tested to see is the Snellen chart. Now, nearsightedness and farsightedness. So many people confuse this. If you are nearsighted, that tells you what you can see. So if you're nearsighted, you can see near, you have trouble with distance. If you're farsighted, you can see far away, but you have trouble with things close up. All right, and a lot of times this starts to show up as you age. All right, your vision is, you think is good, but then as muscles can no longer compensate it, all of a sudden you can see things far away, but up close it becomes what? Really tough. And that's when your arms start to grow longer and longer. Your mom and dad probably do this. Some of them do this, where all of a sudden they're looking at something and their arm is stretched out and it gets longer and longer to the point where they can no longer see without glasses. All right. So far, just remember, nearsighted, you can see near. It's what you can see. Farsighted, you can see far. You have trouble with objects close. Now, what happens with nearsightedness, the object comes into focus where? In front of the retina, and then the rays spread out so it becomes blurry. Farsighted, it's what? Behind the retina, and I think the next graphic will show that. Okay, so as you can see, the candle, all right, is coming into focus or being projected what? Behind the retina, therefore the rays are spreading out, okay? Nearsighted, you can see it's being projected what? In front of the retina, once again, starts to spread out, blurry image. Now, in normal vision, it's the third one. As you can see, the candle projects the candle, that the focal object is the candle. All right, the object, the light waves come in, they're flipped, they're inverted, and it's projected directly onto the retina. So that's normal vision.